and your foot pegging on your accelerator, right? That allows you to go. And if I pull my foot off the accelerator, I decelerate. If I don't have either foot on none of the pedals, you're decelerating. Does that ring home? I don't know how many times, that was many times with different ways I can say it. Okay, that's an important detail. All right, so the idea of an operon. Let's cover that before we hit how those things work in our specific examples. The first idea of an operon is that you have one promoter that's going to make one mRNA. Now that sounds pretty normal of what we've been doing. And if you go back to your exam 1 material, we spent a bunch of time talking about how RNA polymerase, and again, that was one of my first questions today. What does RNA polymerase bind to? Promoter. So remember sigma factor and all that. I won't test you on sigma factors much, but just the concept of RNA polymerase is core to this unit. So to have a promoter is really essential. If I mutate that promoter beyond recognition, will RNA polymerase find it? Okay, so we're going to play a game later that talks about mutation. And if I mutate the promoter and I call it P minus, meaning like a P subscript minus, that means it's damaged goods. And you can never promote it from that. A P plus means it's an active promoter. Okay, so I'm just giving you a heads up that plus minus system works for a lot of things that we do. All right, so you got this promoter. Now you have typically a gene downstream, and you have an mRNA that's made with a +1 site all the way to the terminator. And embedded in here is a start codon and a stop codon. So this would be your coding sequence right here. It's got to make all the polypeptides for that, which makes that the UTR. Still need to know that. And that's the UTR on this side, right? The untranslated region. Right? That's a review from last section, right? Now we're going to bump it up a notch. What we're going to say is we're going to have 3 genes behind one promoter in this particular instance. Really all I have to have is more than one gene behind one promoter. Actually, I'll draw it more simplistically up here, because that looks a little convoluted in that figure, but I'll just make sure we see it. So this would be like gene number 1 and gene number 2. Let's do the simplest operon that we could do. Means you got one promoter, so the RNA polymerase starts. It doesn't stop here because it's designed to go all the way here. So basically, like, there's a terminator right back here. Now, the job of RNA polymerase is not to interpret. It has no care about what the mRNA, or hey, heck, this could be a tRNA right here, or it could be a ribosome RNA, promoter and a gene. It didn't tell you what kind of gene, structural versus non-structural, right? Non-structural meant like RNA genes. Doesn't matter. RNA polymerase in bacteria only has one RNA polymerase, and it's going to do all the jobs, right? Now, whose job is to interpret it is now the ribosome is going to say, oh yeah, there's a start codon there, right? There's where translation is interpreting it, the next level. So let's be clear. RNA polymerase is just yes or no. Tell me how much, and how much do I do it? Do I keep going? Do I make just a little bit? And then you get a transcript. The transcript could be anything. Now in this case, this transcript has 2 genes embedded in it, which means that that gene stopped here technically because the coding sequence stopped. And this one has a new start codon embedded in it, and another stop. Now I almost always test on this concept. Within an operon, however many genes you have is how many start codons you have. It also equals how many stop codons you have. If I have 5 genes behind one promoter, I have 5 start and stop codons. Because each gene is still autonomous from each other, because it's going to make a polypeptide. Now this polypeptide, RNA polymerase is going to start, ribosome jumps on at the ribosome binding site right here, right? It's going to locate the first AUG, and it's going to make that into some polypeptide or some structure. Then, simultaneously, you're going to have another ribosome binding to this ribosome binding site there and making some other polypeptide. So I'm basically, from one transcript, getting two polypeptides knocked out simultaneously, and also, technically, in a polyribosome. Because basically, ribosomes are cranking through here. One comes here, another one joins. So there's a string of ribosomes powering out lots and lots of polypeptide. So it's super efficient, right? It is super efficient at making lots and lots of polypeptides. Now, the reason why they're doing this, so the word polycistronic, which I'm not going to use that word necessarily in a test, but let's break down the word just for kicks. Poly, many. Cis means next to. Tron means new, right? So polycystone means many units that are right next to each other, which is what that's saying. Multiple genes next to each other. Now, here's the kicker though. This is what the most important detail is of like understanding how to bring it together. They do this so they coordinately regulate it. They don't have to regulate two different genes separately on the genome. They can say, I'm going to tie these two genes together so they're turned on simultaneously for a common function. So in our case, the lac operon is about lactose metabolism, to be able to use it for energy. For the tryptophan, it's going to have, I believe, like 5 genes, one promoter, and like 5 genes. And each one of those genes is an enzyme in a pathway to make tryptophan. So they make them all simultaneously. Otherwise, you have to make them hopefully equal amounts from these promoters. And maybe that'll happen. Maybe they'll be unbalanced, right? But that's the kicker, is that you basically need to turn these things on together, right? All right, so let's stop there. Any questions what I've led up to? All right, so now we get to a place where we're going to talk about the details of the lac operon. That's everything's been up to this point in generality. Now, the details of this one, and one thing I don't like about this figure, I probably should try to find another one, is right here after that purple sequence says cap site. I want you to consider that the LAC-I gene is separate and could be very far away from our other genes. I'll redraw it like this. That's okay, I'll do. So the promoter is kicker, that's the key one right there, to making Z, Y, and A. I'm going to shorthand those 3 genes that are sitting behind it. So the LAC-Z is going to be the hardcore one. That's our beta-galactosidase that we worked in the lab recently this week. That's actually doing the digestion, in fact, right here, breaking the galactose and glucose subunits that are connected together in a beta-galactosidase connection, and break that bond, and then you get those to be able to use for energy. All right, so that's essential. The LAC-Y gene is this one. It is the one that allows lactose in. So those simultaneously have to be active. Let's actually imagine if you have this one active, but you have a mutation for LAC-Y, that it's dysfunctional. Frame shift mutation, whatever it is. If that occurs, you do not have that transporter right here. So this is a transporter to allow lactose into the cell. And notice it has an exchange with hydrogen ions, irrelevant for exam, right? But it has a mechanism which it transports it in. Now the deal is, if this is absent, you can have all the beta-galactosidase you want in the cell, and you'll never be able to digest lactose because it'll be sitting out here. You might be like, man, I wish I could have some of that lactose, but it never can get it. So they both have to be working. let's look at the opposite. What if this permease works, and you get it in, and you don't have the enzyme? That doesn't work out either. You get a bunch of lactose in here, and you can't break it down. Both of them have to be working. Now, this one over here is what they call transacetylase, and it basically adds these acetyl groups onto compounds made to detoxify the situation. And we're going to brush over that, because if you lop that off, it still works. The system still works. So the core are these two that we're really going to talk about for the next topic here, for the next little bit. And LAC-A is inconsequential to us, even though a cell has less toxic side effects when it has it. So it's probably helpful, but it's not essential. All right, now notice that's the promoter right there in lime green, and it goes all the way to the terminator. That's your transcript I just drew on the board for you. Now, that is the binding site for the activator. So now we're going to be putting a little flavor about the regulatory elements. And I'll put it as a green. I'll try to, as much as possible, to color code activation as that green equals go. So we call this the CAP binding site, where basically where the activator, the A stands for your activator protein, is going to want to bind to. So that's called DNA elements, right? That's an element, a sequence that it likes. And then next to it, you have the operator. Now, in this case, an operator is really, I don't know why they termed it that way necessarily, but historically, it's basically being said that this is the place where you operate the stop, right? It's basically, so if you have now the repressor bound here, so this would be the activator. If you bind an activator, then you promote the transcription. That was what we just went through. The generic activator binds, let's make it. If a repressor binds, let's stop it. Physically, right in front of where the promoter is, so you can't get by that. That is the functional unit of the operon. Okay? Now, what is LAC-I? LAC-I is the repressor itself. Dot, dot, dot. I'm going to make sure you see that it does not have to be right there. It could be on a plasmid. It could be anywhere in the genome. As long as you make that repressor, the repressor is a protein that goes out in the cytoplasm and cruises around and finds its binding site. This is really important for your next, when we bump it up a next level. Basically, factors that are made into the soup of the cytoplasm. In fact, let's actually do it this way. Here's the cell. Here's the DNA. As long as you make the repressor, and I'll make the repressor like a little triangle. The triangle gets kicked out in here, and then it circulates into the cytoplasm. So meaning it's fluid, it's mobile. How about the DNA element? Can it go wherever it wants to? No. It has no motor, has nothing. It just sits there right here. So wherever this is, it's super important it functions exactly where it is. This gene over here that we're going to call now LAC-I is going to equal to the repressor I for like inhibition, right? So this repressor, when it's made, can circulate wherever it wants to go. So this is irrelevant where it's located. Everyone follow me on that? Secondly, it's constitutively made. Oh, there's that word. Or I basically say not regulated to maybe help you out there. If this is not regulated, it means you make out a steady state amount of repressor. You're always kicking it out. That's like your smoke detector. This is your repressor that's basically like, I'm out here, okay, I'm checking out what's going on. Tell me when I'm supposed to bind, right? Now, there's another gene somewhere else. In fact, I'll go downstream of it. Dot, 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 somewhere else. you're going to have a promoter, and you're going to have the cap gene. And a cap is not regulated. And it turns out it's your activator protein that's made. So this is what we call your activator. Now, why is it called cap? It's called catabolite activator protein. And the catabolite, you're going to find out later why it's called catabolite, because it's actually subject to catabolites or metabolic breakdown products. Or in other words, cyclic AMP is going to be its story. So let's not worry about the word C on it. Just think about activator for now. Okay, so guess what? The activator and the repressor are both sensors that are sitting in the cell and need to be present. Now, they don't need to be active necessarily at this moment. They only need to be active when they're supposed to be active. So what's going to activate them or inactivate them is an effector, right? Back to what we talked about. So they're made, and then there's rules to their game. Now, both of these situations is an inducible scenario. Just like we did in lab, we induced the lac operon to turn on. That yellow color you saw in the tube was because the lac operon got activated by giving it lactose. Not only did we give it lactose, but we didn't feed it any glucose in that same tube. I want you to remember, when you set up 4 tubes, one had glucose, that's it, about the media and stuff, and the other one had lactose, no glucose. So in essence, if it doesn't use the lactose, it's starving, right? So it's an inducible situation, which makes it turn that yellow color because we could sense how much beta-galactosidase was there through our assay, right?
So since the binding site is kind of towards the middle, but the repressor and the operator are on opposite ends, does it kind of work like a magnet, like it wants to pull it?
So basically, what I said is ... means like, this could be where the lac operon is. This could be where the cap is. This could be, they could be, it doesn't matter where they are relative to each other. It doesn't matter. This just means somewhere else. Somewhere else. I'm not trying to influence you to think one's upstream, one downstream.
Okay. ...
means I don't care where it is. In fact, I could put this on a plasmid. Now, is a plasmid in a cytoplasm? Transformation, remember that? Yeah. So if I have a plasmid right there, when I kick out proteins, it goes right into the cytoplasm itself too. Now, why is that important? Because we're going to play a game, and I'm going to call the IPOSI later. It's going to be on a test, on your homework, and it's going to be understanding that these proteins can come from anywhere you want. In fact, imagine this. Let's say you took away this gene, and you're like, oh man, I'm not going to have an activator. And what happens if you had a magic little syringe, and I punched a hole in here, and I popped it in there? Is it present? Yeah. I don't care where it came from, as long as I have it. Does that make sense? So I don't care where it's made in the genome, and I'll tell you what, this unit is the operon itself. These are regulatory genes that make the regulators themselves. Okay, everyone with me? Take a little pause right there. So that's super important. These are the genes that actually do things for the business end of lactose metabolism. These are just telling it when to turn on and off. That's what you call regulation, right? Regulatory genes. Okay, so here's what's interesting about the LAC-Z gene. The LAC-Z gene can either break this bond right here, and then we can metabolize. The glucose goes right into glycolysis. The galactose gets turned into glucose through another enzyme that actually converts this site right here called an isomerase. So basically, you get 2 glucose units out of this for ATP because when you break these down, both sugars can be used eventually, right? Now, this is an alternative formation of a side reaction, as it's called. And it makes only a little bit of this. That's not its primary goal, but it makes a little bit of this, and it'll break down this. So when it makes this structure right here, we call it allolactose. Now, the really critical thing about that word What did I say what allo means? Other. Allo, let's go back over here. Allosteric. Other structural binding site. Other. Allolactose. In this context, other formation of what lactose looks like. So it's going to make a weird bond out of it in an other form of it. So we call it specifically allolactose because this bonding structure makes it unique. Now, allolactose is the effector. It is the effector. So when the presence of allolactose happens, it will bind to the repressor itself and help pull it off. That'll be your first message that's important. Lactose talks to the repressor. How does it talk to the repressor? Through this structure right here. So the repressor can't bind to lactose, it doesn't bind to any of this. It only has a little binding site, that effector, that little dip, for this molecule. And if it's present, the repressor will release from the binding site of the operator. All right, so let's actually go back to the car analogy before we get too far into the weeds here. When you start your car, what status is it? Is it going to be in a go status or is it going to be in a brake status?
Brake.
Brake. Because if you turn on your car and you start going, you're going to run into someone. Right? So even right from the get-go, you always have the brake on. That's exactly what's going to happen here. This repressor is always going to be on there. I shouldn't use the word always. It will be on there from the starting place of saying, let's start off in the stop position. Now, if you have the presence of lactose, some of that will get converted into allolactose, and what will happen is it will release, because it's saying, oh, we have lactose, or the allolactose, because some of it will get converted, and then it'll come off. And when it comes off, then RNA polymerase can go. The thing is, just pulling your foot off the brake doesn't make you accelerate. Right? In your car, if you pull your foot off the brake, you're not going anywhere. Your car might actually go this fast, like in idle, but you're not going to go freeway speed, right? So you physically have to put your accelerator down to actually have cap bound. So cap, this right here, actually I'll write it this way. Responsive to lactose presence. It's sensing lactose. It does not care about glucose levels. I don't care. So really the repressor says on, off, relative to how much lactose is around. If there's no lactose, shoot, lock it down. There's no reason to turn this on. If I have lactose, pull it off. Right? Now the activator's game is different. The activator is responsive to glucose. And it goes in the opposite kind of way. So this one says the activator is going to be not active when you have lots of glucose. Okay, so basically the rules to this game are such that do not put the foot on the accelerator and make it go fast if you have lots of glucose and you can use that in glycolysis already. So you need to be starving. You need to be lacking glucose, which means low ATP levels. And if that's true, you want an alternative sugar source because you're basically dying, really. You have no more energy. So you start going, oh, if you do have lactose, the repressor will be off, right? Let's start back over here. You have lactose, the repressor's off. You're starving, so you put your foot on the accelerator, and that's how you drive to work, right? You have to pull your foot off the brake, put your foot on the accelerator, and now you can go. If you put your brake down while the accelerator is on, is that going to work out well for you? No. So the only way you really get to work on time is you pull your foot off the brake and you hit the accelerator. If you just pull your foot off the brake, that's not going to work out well for you either. So what we're going to do is follow the scenarios, and they're basically, the reason why this problem is a little complex is you have to break the problem into its parts, which actually worked out well that they're on opposite sides. The truth is, I can only talk about the activator with respect to glucose. I don't care about what's back here. I don't care about lactose. And then when I say I'm going to talk about the repressor, I'm going to flip and I'm going to turn and say, I'm only talking to lactose right now. I don't care what's going on with glucose. And those are your pedals. That's what happens when you put your foot down on one or the other. So imagine if you drove a car with 2 feet. I think some of the go-karts, like you go into those little go-kart places, you have to use both feet. It'd be more like akin to that, right? You use both feet. Now, pull both feet off, you're not going to go anywhere, right? So that's the analogy you need to look at. Common sense driving, and the only way you get to where you're going is pull the brake off and accelerate. Okay? All right, so the DNA elements, or what I wrote up here, regulatory elements, I guess I got rid of them, are the promoter, the operator, the cap site. Those are all what's regulating the system. Then the structural genes, and if you remember what structural means, those mean polypeptides, right? Non-structural genes means RNA. So there's again what I just told you. LAC-Z cleaves it or forms allolactose. LAC-Y allows it to come in. LAC-A, who cares? Don't worry about that. All right, so first let's talk about the repressor only. And then we're going to switch over to the gas pedal. And then we bring them back together. We're going to actually, so here's what you have to do when you solve these problems. Find out what's going on with the repressor. Find out what's going on with the activator. And that equals your driving pedals. And then that equals whether you can go or not. Like what you're doing at the pedals. So the first one is this weird looking repressor. I kind of watch too much kids stories, or Lorax kind of always comes to mind when I look at this. It has like little feet right there. It's actually working as a tetramer. That's 1 subunit, 2, 3, 4. That's not relevant for you to memorize. But notice it has a little spread that actually touches different parts of the LAC-O DNA sequence. So it's kind of covering it up. Now, those are its effector binding sites, the little notches right there. Now, right now, since lactose is not present, it's in the binding position of like being an active repressor. Meaning it's actively or basically wants to bind to its binding site. So that's its like first initial impetus, right? So basically right now it's on lockdown. Now LAC-I is being made constitutively. It's being shown properly here. You're making constant amount of the LAC repressor, and the word active only refers to its binding activity, right? So that's when you do not have lactose. Now here's the story. Do not turn on the gene unless it's present. Why would you start trying to make all these enzymes when lactose isn't even there? That's again common sense mentality to say every single one of these is going to cost you energy to go through translation. And 3 polypeptides is going to be a considerable amount of investment. So don't make those, and that's what I said. It's very efficient to block transcription and not make polypeptides if you don't need them. So that's what we're doing here. It's very core to the situation. You don't have any lactose? Oh, well then just don't make these, right? Simple. Now, when you do have lactose present, now this is where it gets a little interesting about this too, that you think about it. You have to have just a little bit of the permease to allow some lactose in. So we're going to call the system a little squeaky or leaky, and that every once in a while the repressor will come off. Let me just go back to this. And this is not covalently bound. Let's actually remember actually let me bring up a concept from biochemistry that you may have heard or may have not. But it's called the relative affinity of binding, the binding affinity, in other words. So an antigen and an antibody. When an antigen comes in the system, you have antibodies that recognize it with a really good strength of binding. They'll lock it up and then create a reaction of like immune response, right? It's one of the strongest binding interactions. So that's a pocket, and it binds to it very strongly, but it's still not covalently bonded. But it has a pretty strong binding site. Whereas other ones have a kind of a looser one where it's going to bind, but it comes out at a higher frequency. So the higher the concentration of it, the more of it goes into the pocket and keeps it occupied. So low affinity means you just have to have more of it, and it'll keep going into the pocket. But a high affinity means it's going to hold it and hold on to it for good. And the reason why I bring that up is because even if you had this right here, even this binding site with DNA is going to release at some point. It's not covalent. And guess what? One of these transcripts is going to slip through. And that's going to be like the basal squeaky level. Now, why am I saying that? Because right here, that is the basal level right now. Lactose permease, the permease is present at a very low level. And the reason why that's important is because when lactose becomes present, you have to be able to get it into the cell, at least a little bit of it, to start getting the whole system going, right? Because if you didn't have any permease at all, lactose would never get into the cell. And then beta-galactosidase, which is one of these, okay, that one right there, needs to convert it to allolactose, and then that becomes the effector. So you can't start the chain reaction unless you have a little bit of it, right? So that's what I call squeaky. So let's go back to the slide then. That's the second one. All of a sudden, lactose becomes available. You have a wee little bit of permease that allows the lactose in. Beta-galactosidase, we have a wee bit of that, and it basically will make some allolactose. Now, this is going to prime the system to like get it going. And what's going to happen is it's going to bind in those little pockets, or let's imagine it right over here. Allolactose now is floating around, and it's going to attach, 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 and attach. And then what happens to the feet is they release or they change conformation. So you remember when you have like something that comes into a binding site and it causes a conformational change? Allosteric? That's what's happening. So when it hits it, it's going to make it fall off, and then it's like this. So this would be when it releases from binding. Now what happens now is it's permissive, and I'm going to use that word very specifically. Permissive means it's going to allow RNA polymerase to come through at a very low rate. At a very low rate. Because that's when you don't have an accelerator. If you basically pull your foot off the brake pedal, your car will probably go maybe idle, like 2 miles per hour. And if you're relying on that to get you to work, that's pretty sad, right? That's not going to get you there. So that's where we have to flip our discussion later to the activator. But right now we're focused completely on the repressor. And if we didn't need an activator, like if you're in a car where gravity, I just pull my foot off the brake and I just go, then that would be a scenario in which this would work well. You just pull your foot off the brake and you go. But that's not what's happening here. You have to hit an accelerator. Okay, so it's as simple as when lactose is not around, it's bound up, and you can't use the RNA polymerase, or at a very, very low rate, because remember, every once in a while it'll come off, and that gets you here. Then lactose becomes available. And now you're going to look at the system get primed up. Once a little lactose gets available, it's going to pull the repressor off. Notice the arrow. And then you ramp up the system, and you're making more and more and more of it. And now just like, let's just say that activator's involved here, because this is a very simplistic model that's not incorporating the activator right here. It's just saying this would allow you to ramp, be permissive to do this. And then you get lots of permease, you get lots of beta-galactosidase, You finally digest all of the lactose available, and then you have all of these proteins that start down-regulating, such as they get recycled as proteins digest, because they don't live forever. They have a half-life. So you have to keep making it over time, because eventually they will degrade, right? And then you return back to this. So when you have no lactose, it'll take a little while, but eventually the proteins will return to a steady state, very basal level. All right, so let's pause right there. Questions? So really, it's just where you pull your foot off the brake, right? Okay, so this is going to get us into, it looks like we have enough time to start this exercise and maybe actually finish it. So what we're going to do is talk about a scenario in which bacteria have 2 copies of the lac-I gene. Now how this happens, they call it a merozygote. You don't have to know that word, but these individuals discovered that a plasmid can contain the lac operon too. So what that means is we got this complexity. This is the problem that we're going to talk about. All right, so let's. So that's the lac operon, and then here's your plasmid. I'm going to draw the plasmid a little bit bigger, just so I can draw the lac operon on here. So you have 2 copies of it. Now, you're not called a diploid in this context of having 2 copies of every gene on the genome. You're called a partial diploid because I have 2 copies of the genes that the plasmid has that the chromosome shares. And in this case, we're only focused on the lac operon. All right, so what we call this is the I-POSY example. Okay, so the I-POSY, you can have it from the chromosome. And that makes a very simple story because I don't have to complex it with this. So first we're going to start off talking about that. And then we're going to add the plasmid to the situation. And we're going to have 2 copies. Now, why is it called POSY? This is the lac-I gene, which is separate. It is separate and constitutive. This is your unit of the operon that is promoter, operator, and ZY lopping off the A. Now, anytime I write a positive on it, it means it works. If I write a minus on it, it means it's dysfunctional and you can almost erase it, just like remove it. I minus, or say if I have I minus, that means you have absolutely no repressor made in the system. You can't make it. It's impossible. It's been deleted, damaged goods, frame shift mutation, you name it, whatever its problem is. Now, if I have a minus on the operator, that means you delete the operator, let's say. Now, where's the repressor? Tell me where the repressor binds. At the operator. And if you have no operator, there's no repression. Right? So here's the deal. If I have I minus and O plus, that's not going to work out because you don't even have a repressor to bind to it. So I want you to think about your foot is the repressor in the car. That would mean you don't have a foot. That's kind of sad, but you don't have the actual ability to push on the brake pedal, right? Now, this would be like the brake pedal's gone. You're like, where is it? It's not there. And then you're just going to crash, right? So this is what we're going to call a factor or a protein that can go anywhere in a cell. If you remember back to what I was talking about, when a repressor is made, I can go wherever I want. So it's not like on lockdown at all. It's like once it's in the cell, I can go where the heck I want. Now this operator can only talk to that promoter. That's what we're going to call the cis effect. In fact, let me put it on the next slide. The cis effect, might as well start, well, let's go back to what I just said. The trans effect means any protein that's made that can move anywhere once in the cell. And that pretty much is any protein that gets secreted, put into the cytoplasm. So once it's out there, it can actually do what it wants. Now, the cis effect or cis acting type things means that this operator, it's a DNA element, in other words, AKA, it means that this thing has to be exactly where it is, and I can't put the operator over here, move it, and it won't block RNA polymerase. I can't move it over here. And it also won't talk to this one over here because there's physical separation of where the plasmid and the chromosome are. So cis means it has to be functioning exactly where it is. All right, so what the game here is how to dissect a system with how it regulates. And then there's a couple of lessons to be learned in this. Now, OC, I'm going to be writing it as an O minus, so you can write that down in your notes. And in the literature, it's called C, but it has the same effect of just saying O minus, which means it doesn't exist. It's obliterated. It's deleted. All right, so this is where we're going to get to. We're going to have an IPOSI set, but before I launch you on this one, we're going to do a similar one. I'm going to take this one off, and we're going to say, this is the presence of no lactose, and this is in the presence or having lactose. Like you do have it, positive, right? Okay, so what you're going to basically say is whether you make the transcript or is the operon on or off in these situations. Okay, so go ahead and talk to your neighbors if you want. And this is not going to be an iClicker version. This is just between us right now. Okay. Didn't hear anyone chatting it up, so I guess you're ready. Here's the key, think about it this way. This is about logic flow. If I do not have lactose, what will the repressor be doing? I'm going to have to ask for someone.
It's going to be off.
It's going to be off. Because the repressor is going to be bound to the operator when you don't have lactose. That's what we just all talked about, right? We're all going to put a down arrow, meaning it's off. It's being repressed. When lactose is present, what will happen to the repressor? There you mouthed it. It's going to come off because allolactose will be made, right? And then it's going to pull it off. We can just scroll back a couple slides, remember? So we upregulate. So basically, this is what's supposed to happen. When everything is working properly, when you don't have lactose, you shut it down. And how do you shut it down? By the regulator. The regulator is this talking to this. And both need to be present and working. If one is dysfunctional, then you have like a foot trying to push on a pedal, or you have like, no foot being able to push, right? That was what we were talking about. So this is what's supposed to happen. Now, I'm going to kick this to you now and see if you can figure out what happens when the operator's damaged. Okay, spend a little time with that. Talk to your neighbors if you want. Not a bad idea.
They're going to do the same thing. Yeah, right? So that means they're both off? Yeah, okay. Do you you into paying attention anyways?
I was doing this section because I saw this, so I got scared.
There's gonna be like 2 separate videos, by the way, because my I just turned off for a second. I was like,
That's okay.
All right, so I'm going to give you a countdown. Even if you don't understand it quite yet, we're going to have multiple chances at this. This is your first one. All right, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Actually, let me look the right answer up there again. Okay, there you go. Yeah, right. Just double check. And it is A. Okay, if I say the word constitutive, what does that mean? Always on. So O minus is the same as OC, right? Okay, so here's the deal. What we're saying in this example is I'm taking out the operator. That's what O minus says. Remember I told you? Just delete it. So now let's look at that again. If I gave you that example on the board, can you, the repressor, stop the promoter from working? No, honestly, answer me back. Can it? Is there anything blocking it? No. So what happens? RNA polymerase binds and it just makes it all the time. It's not regulatable. We just took away the operator, the binding site where the repressor is supposed to go. So that means it's on even when you don't have lactose, because you're not regulating it with lactose anymore. You're just turning it on all the time. Everyone with me? Okay, I'm going to give you a second example without the iQuicker. Okay, do that one. Just take it as seriously as the last one of the iQuicker, like try to see if you would answer it. Like it would be the same options up there.
The I is what makes the regulator. Well, it makes it. The lac I makes it, and then that ends up binding up here. So that's the actual
What happens
repressor.
if I do this?
See?
Minus means it's gone.
I can't make it.
You can't make the repressor, so it's just always going to be on.
Doesn't work. Minus means it's dysfunctional. Okay. So what do you think? Did that change the answer?
No.
No, because it's not regulatable. You have to have a repressor to push on the brake pedal, which means like it's your foot not pushing on it. And if the pedal doesn't exist, then it doesn't work either. So it's hand in hand. They work together. All right, so here's the first thing you need to solve these problems. Number one, does it have a promoter? Because what happens if I do this? What will that do? I heard you mouth something. RNA polymerase can't bind it. So what you have to think about is what is the purpose of a promoter? And we started off today talking about that. The promoter is found by RNA polymerase to be able to start the transcript. And if I told you the promoter's gone, you're never going to make anything out of it at all. So you stop the problem if you don't even have a good promoter. It's done. I don't care what else you draw. The promoter has to be there. Okay, the second thing you do to solve this problem is ask yourself, is it regulatable? Now, regulatable means both the interaction of the I and the O, right? That you have to have a plus. Somehow, to make these things two work, you have to have both of them somehow working, right? You have to have a brake, and you have to have a repressor. Does that make sense? Okay. Okay, so now we get a little more complicated. Now we're doing two different gene transcripts over here. Let me see if this one. Yeah, this is a good one. All right. So when I throw a plasmid on here now, I get the same output that I'm putting on here to here. Now, the way we're always going to play this game is only one of these two is functional from the one, and this is the opposite. In other words, they have to complement each other, or basically saying plasmid can only make the permease, because I can like erase the beta-galactosidase when you have a minus, right? And it means that only beta-galactosidase is important from this transcript, and this one's damaged goods. Okay? So this is the next layer. Okay, so basically, what you're saying is, you've got this scenario which all you got your pluses, now they can start talking to each other if they're trans, because trans means that they can move around in the cell. But if they're cis, which means that that's what we're going to talk about, have to be exactly where they're at, then they can't help out the other one. All right, so let's try this one.
B-gal is Z and Y is permease, right?
What's that?
B-gal is Z and Y is permease, right?
Right.
Okay.
Beta-gal, Z. Yeah. Okay, first of all, I'm just going to direct your attention to the method to the madness over there. First, you have to see if the promoters work, right? Then, secondly, you have to decide each of these transcripts that are going to be made, whether they're regulatable. Okay, and then you're going to answer your questions, right? Those are the steps you take. All right, we're going to go at that 10-second count. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. All right. A chunk of you got on that one, and some of you missed it, but now the majority picked up. Because you don't have any repressor at all in the whole system. Didn't we? Isn't that just like what we just said? If you don't have any repressor, it's all relevant. This one's going to be made all the time, so you're always going to make this one. And this one over here is always going to make this one. It's going to leap over this one. It's like deleted, right? So basically you have no repressor. So it's not regulatable for either transcript, so they're always on. Right? Okay, let's try the next one. Okay, here we go. So, erase, right? Okay, but I'm going to ask you too, before you finish answering this, or maybe you already answered it. Can the repressor only talk to this side over here, even though it's in the chromosome? Didn't we say the repressor can go wherever the heck it wants? I want you to think about what that means. It's in a trans effect. It's the trans that can move wherever it wants to. There's no blockade, right? So, can it talk to this one? All right, we got a couple minutes left in class. We're at 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. All right, bunch, you got it. It's D. Okay, let's isolate. Okay, here's the deal. This is called problem solving 101. Meaning, I'm going to first talk about this one. Let's first just solve beta-galactosidase, because that's the only thing can be made from up here. It's the only thing that's relevant when we talk about the lac-Z. Now, since the operator's broken, it's immediately on all the time. You can't solve it with the help from this one. That's called cis effect. This one has to be exactly there. It has to be functional. And if it's gone, it's going to make it all the time. So with respect to beta-galactosidase, on and on is the right answer. And you just answer that part of it. It's not relevant to permease. Now permease, I'm going to talk about this one, because that's the only one that can make this. Now, I do have a good operator. By the way, both promoters are functional, so we're good there. But this operator works. Now, I look at this and I go, I don't have any repressor here. That means you're not being able to stop it. But I can get it from here, because once it's in the cytoplasm, it can go wherever it wants. That's the trans effect. So basically, this one will now put it on here when there's no lactose, so you don't have any permease until lactose is present, and then you pull it off, and then you make it. Now it regulates. Not regulating means you make it all the time. All right, we'll come back to it on Thursday for a brief couple more minutes, and you're going to have to just spend a little time with that.